like my students could possibly benefit from some notes on cellular respiration. So hopefully this helps at least one person. But when we look at cellular respiration, the big idea is that we are trying to harvest energy from our food, our organic molecules. So our organic molecules could include carbohydrates, lipids, or proteins. Now, when we learn cellular respiration here, though, we're going to be using glucose as our model for understanding how aerobic and anaerobic respiration work. Now, both of these are metabolic pathways. When I say both of these, I mean anaerobic and aerobic respiration. So, metabolic pathways are pathways that use enzymes. So these are use enzymes. So metabolic pathways use enzymes in a series of reactions. So when we look at cellular respiration, we basically are going to start with glycolysis, and at the end of glycolysis, we end up with pyruvate, or pyruvic acid. And so we start with glucose up here. And so there's about 10 different reactions that we'll talk about. Now, throughout the process of glycolysis, though, the initial stages require ATP, but really one of the steps takes NADH and reduces it to NADH. Now, if there is oxygen present, after glycolysis, the pyruvate will enter into the mitochondria to be further oxidized to get any remaining potential energy. However, this is if there's oxygen present. If there is no oxygen, if oxygen is not present and glycolysis is the only way that that cell is producing ATP, we need to make sure that at least glycolysis can continue even in the absence of oxygen. So the purpose of fermentation or anaerobic respiration is basically to keep NADH becoming oxidized to NAD+, so glycolysis can continue. Like, that is the main purpose of fermentation. And when we look at aerobic respiration, pyruvate has 3 carbons, and there's still potential energy in these bonds. So that is why in the presence of oxygen, we can further oxidize this in the mitochondria. So at the end of aerobic respiration, there will be a whole lot of ATP produced at the end of aerobic respiration. So those are 2 metabolic pathways following glycolysis. So in reality, we have a couple different steps that are possible. So let's start with glycolysis first, and then we can move into fermentation, and then we will discuss aerobic respiration. So in glycolysis, we are going to start with a 6-carbon molecule. So this is glucose, C6H12O6. Now, at the end of this, we will end up with 2 pyruvates. So in one of the very first reactions, there's actually 2 ATP are required. So when you see this arrow, like here, we're going to go down in a series of steps. When you see this arrow coming in, that means that this is required and this is the product. So we end up actually investing 2 ATP in this first step. 1, 2, 3, 4, 5, 6. And we actually will put 2 phosphates onto that 6-carbon molecule. And then another enzyme. So every time we do an arrow like this, that is implying that it's an enzyme-controlled reaction. So here, our next step is an enzyme will actually break this 6-carbon molecule into 2 3-carbon molecules. Then in our next reaction, what we're going to have is we're going to have NAD+, which is an electron carrier, is going to become reduced. And this actually happens 2 times. So we have 2 NAD pluses become reduced to NADH. And in this bond right here, there are some electrons. And when we move electrons in a cell, that is how we're moving energy. So right here in this step, it's picked up some energy here that will be used in the electron transport chain of aerobic respiration. Now in this step as well, we have 2 inorganic phosphates invested. So we end up with a 3-carbon molecule that has a phosphate group on each side. And then in a couple more reactions, what we're going to have is we're going to have some ATP produced. So again, 2 ADP. So when we look at this, basically, what we see here is that, oh, I don't have a highlighter with me, but what we see here is that this phosphate, let's see if I can write on it with a pen. These phosphate groups, like here's a phosphate and a phosphate, those are what's added to these 2 ADPs to make these 2 ATPs. So here we had 4 phosphate groups, and now at the end of glycolysis, we end up with 4 ATPs. But we need to remember that at the beginning, we actually invested 2. So our net gain is basically 4 ATP minus 2 ATP for ending up with 2 ATP. Let me check my drawing, sorry. Okay, so I think I was saying something about the ATP. So your net gain of ATP at the end is 2. Now, some other products that we have that were made during glycolysis is these down here are pyruvate, but sometimes they're also called pyruvic acid. So either one, they both mean the same thing. And if you notice how we started with the 6 carbons up at the top in glucose, we now have 2 3-carbon molecules. So when we look at our overall process of glycolysis, it is a metabolic pathway, and it's a series of steps of enzyme-controlled reactions that initially is endergonic, which means energy requiring, and then we have some exergonic steps down here where we end up producing 2 ATP molecules, or a net gain of 2 ATP. We also get these 2 reduced electron carriers, NADH, and we end up with 2 pyruvates. Now, the process of making ATP, so when we take ADP plus a phosphate to make ATP, we call that phosphorylation. However, so when you add a phosphate to something, that's called phosphorylation. So in glycolysis, the process of making ATP is substrate level phosphorylation. Because we got the phosphate off of a substrate in order to make this ATP. Okay, so other key points about glycolysis. It occurs in the cytoplasm of all cells, and I wouldn't classify it totally as anaerobic, but it happens without oxygen, but also occurs in aerobic respiration. If there's oxygen as well, it happens before either fermentation or aerobic respiration. So it happens first, and it produces 2 ATP, which is a small amount. It's also like evidence for common ancestry. Like it happens in all cells. There's a good chance it's evolved before photosynthesis, before there was a lot of oxygen in our atmosphere. And yeah, so that's my notes on glycolysis. Now, we might as well just talk about fermentation real fast while we have this here. So when we look at anaerobic, in anaerobic respiration, it's the source of energy is ATP from glycolysis. So in anaerobic respiration, these ATP that were produced, that is your source of ATP. So you want to make sure, so if we have our glucose, C6H12O6, and at the end, we end up with pyruvate. Well, we have two of those. We can put them both here like this. We end up with pyruvate, and along the way, we ended up with 2 ATP as a net. I'm just going to write the net gain, but we also needed 2 reduced electron carriers are produced. So now, in order to continue glycolysis occurring, what is going to happen is this NADH needs to be oxidized. We need to get the electrons and the hydrogens off and basically get it back into NAD+. So NAD+ can continue being used in glycolysis. So at the end of glycolysis, pyruvate, if it's aerobic, if there's oxygen, will head into the mitochondria. However, in anaerobic, without oxygen, in anaerobic respiration, pyruvate is not used. So in this reaction, this chemical reaction right here, the electrons and hydrogens will be added to pyruvate, and that is going to convert pyruvate into lactic acid. So lactic acid is still 3 carbons, but there's a chemical reaction that occurs in order to oxidize this electron carrier, forming lactic acid in lactic acid fermentation. So this is like a waste product of anaerobic respiration. There's still potential energy in these 3 carbons, and what will happen is your liver can actually convert this back into pyruvate, so you don't waste that potential energy. Now, this occurs in like our muscle cells and some other organisms. However, there's another option that occurs in yeast and some prokaryotes, which is alcoholic fermentation. Alcoholic fermentation, and in alcoholic fermentation, the purpose is still the same. We're going to take the 3-carbon pyruvate that was produced, and in order to oxidize this electron carrier back to NAD+, so glycolysis can continue, the pyruvate is actually converted into an alcohol called ethanol, and then carbon dioxide. So in alcoholic fermentation, it's still the purpose of oxidizing this electron carrier back to NAD+, but in that chemical reaction, you actually get an alcohol produced. And that's where like how wine and beer is made, is with this alcoholic fermentation. Yeast do this, and so the carbon dioxide gas that's produced here is what causes your bread to rise when you let the yeast sit in your flour for like your dough an hour or so before you bake it. Okay, so that is anaerobic respiration, and both glycolysis and anaerobic respiration are happening in the cytoplasm. So to summarize, we go back to our original notes. We did glycolysis, and we ended up with 2 pyruvates, but in anaerobic conditions, we need to oxidize the electron carrier so that glycolysis can continue to make the 2 ATP. Because right now, that is the only source of energy for that anaerobic cell. Now, in the presence of oxygen, however, we're going to move into the mitochondria and look at aerobic respiration now. So, in aerobic respiration, this is occurring in the mitochondria, and it requires oxygen. So, you could write that if you want. Occurs in the mitochondria, requires oxygen. Okay, so we're going to start out with our pyruvic acid, or our 3-carbon molecule, and our first step is we need to oxidize it. So So we're going to oxidize pyruvate. So our first step is the oxidation of pyruvate or oxidized pyruvate, and this is occurring in the mitochondria. And so what's going to happen is an enzyme will do this, but basically we're going to have a chemical reaction come where we're going to take an electron carrier, NAD+, will become reduced to NADH, and in that process you're left, one of the carbons comes off as carbon dioxide, and we're left with this 2-carbon acetyl. So now we've picked up some electrons from the pyruvate. So we oxidized the pyruvate. We basically broke this bond, this covalent bond here, and now the electrons are in the electron carrier. And in the process of breaking off that bond, this carbon now has left as a molecule of carbon dioxide. And so when you exhale, this is the carbon dioxide you're breathing out. All right, so now we have this electron carrier reduced, and we have this 2-carbon acetyl. Now there's a coenzyme A that's going to come and attach to this acetyl. So here we have acetyl-CoA. So acetyl-CoA is going to enter into the citric acid cycle. So this happens first in the mitochondria, and you notice there's no ATP produced here. This is not an energy producing pathway. It's just the beginning steps so we can enter into the citric acid cycle. So here we have our 2-carbon acetyl, and that coenzyme A helped it enter in, like it helps it, co means to help. So here it actually is going to join with a 4-carbon molecule called oxaloacetate. So to start the citric acid cycle, we have here a 6-carbon molecule. Now, throughout the Krebs cycle, our purpose is to get any potential energy that's remaining in these 2 carbons, like in the bonds that are still here. So we need to fully oxidize our food. I don't know what to write that. The goal here is to fully oxidize our food. Get any potential remaining energy out in the form of reduced electron carriers. So here, an enzyme will come, and we're going to take another electron carrier, NAD+, and we're going to reduce it. Now, in that process, though, this carbon will leave as carbon dioxide. So that's another one that you are exhaling. So then our next enzyme-controlled reaction, we're down to 5 carbons. And now another electron carrier is going to come in, break those bonds. An enzyme will do that. Break the bonds, take the electrons. This carbon dioxide, this will leave as carbon dioxide. Now we have a reduced electron carrier. So we have this NADH, this NADH. Now in the next steps, I'm going to just draw some arrows to represent like a cycle. In this process, we get one molecule of GTP, which is very similar. It's also a phosphate molecule to ATP. Then our cells will actually convert this into ATP. So we get one ATP made by substrate level phosphorylation. Okay, and then as we continue, our goal now is really to get back to the beginning of the cycle where we had that 4-carbon molecule that we started with. So along these different chemical reactions, we're actually going to get a few more electron carriers reduced. Now, one of those electron carriers we haven't seen before, and that's FADH2. So FADH2 is also an electron carrier that will carry electrons over to the electron transport chain. So now let's think about this, guys. At this step, this looks kind of messy, but here we have everything leading up to the electron transport chain. So if we had a cell, and in the cell there was a mitochondria, this would all all be occurring within the mitochondria. Now, the citric acid cycle happens in the matrix of the mitochondria, which is this center part here. Okay. Now, the other thing I want to point out are all of these reduced electron carriers. This all came from just one pyruvate that we got from glycolysis. But if you remember, in glycolysis, we actually make two pyruvates, or two pyruvic acids. So in the counting, we have one electron carrier reduced, 2, 3, 4, 5. That's going to happen twice because there were two pyruvates. So at the end, we end up with 10 reduced electron carriers. You don't have to remember that, but that's a lot, right? And if we go back even further to glycolysis, we had 2 here. So basically, from one molecule of glucose, we end up with 12 electron carriers. Now, why I mention those electron carriers is because, sorry, I'm using my kids' notes. The electron carriers is because now, as we go into the mitochondria, and we look at like this electron transport chain, here's the matrix. This is where that citric acid cycle was just occurring, where we had all of those reduced electron carriers all in this space. So when we look at the mitochondria has 2 membranes. It's a double membrane bound organelle. And the cool thing about the mitochondria is that it evolved from a prokaryote millions of years ago. So this outer membrane is a lipid bilayer, just like you would find in a cell membrane, which makes it impermeable to ions because of that nonpolar center. Same thing here. This is also a lipid bilayer, and this is important for helping to establish that proton gradient. So here, the mitochondria is surrounded in the lipid bilayer, as well as the inner membrane of lipid bilayer. And so in this inner membrane, we find the electron transport chain, which are these proteins here. Now, at the end of the electron transport chain is this, I shouldn't have written it there, sorry. I don't have whiteout. Okay. So there's oxygen at the end of the electron transport chain. I really should have written it like a little bit closer. That's okay. So here, we're going to take these reduced electron carriers, where we just made a whole bunch in that citric acid cycle, and we're going to oxidize them into NAD+. Now, that hydrogen is going to get pumped up into this intermembrane space, and the electrons are going to get pulled down the electron transport chain, where at the very end we have oxygen is the final electron acceptor. It's very electronegative. So it's very electronegative. It's like pulling those electrons down the electron transport chain. And at the end, the electrons join to the oxygen. Now, as we saw before in like the glycolysis and oxidation pyruvate, when a molecule gains an electron, it's also going to gain hydrogen. So as we oxidize these electron carriers that we produced in earlier stages, the electrons continue to get pulled down the electron transport chain, and protons get pumped into this intermembrane space. When you have a lot of protons in an area, that gives an area a low pH. So if you ever are asked a question dealing like in regards to pH, like the intermembrane space is going to have a lower pH compared to the matrix. Now that difference in pH is like a concentration gradient. There are more protons. This is called the intermembrane space. There are more protons in this intermembrane space, and over time, like they're going to end up flowing through this protein here, which we call ATP synthase. And ATP synthase is going to take an ADP plus an inorganic phosphate, which means a free it'll phosphorylate them into ATP. So, as the flow of these protons flow through ATP synthase like turns, like a turnstile, and in that turning, it like joins the phosphate. It does the phosphorylation to make this ATP. But you need the flowing of these protons in order to turn ATP synthase. So now you think to yourself, okay, well, what happens to those protons as they flow into the matrix? Because wouldn't it eventually reach equilibrium? Well, that's where oxygen, as the final electron acceptor, it actually becomes reduced. It's going to gain those electrons and hydrogens, and it's going to form water. So when we look at our formula for aerobic respiration, C6H12O6 plus 6 oxygen gives us 6 carbon dioxides and 6 water. So the 6 carbon dioxides we saw in being produced in the oxidation of pyruvate. We had carbon dioxide produced, as well as carbon dioxides produced during the citric acid or the Krebs cycle. But the water comes from this oxygen here gaining the electrons as well as the hydrogens to become reduced. Now, if for some reason you stop breathing, then there's no longer an electronegative final electron acceptor, and the electrons do not travel down the electron transport chain. And if they're not traveling down, then there's no proton gradient, and ATP production stops. And this is why death results in like suffocation. And what else can I tell you? Yeah, I think that's it. So basically, so if you want to see the word chemiosmosis, chemiosmosis is the diffusion of ions. Oh, no, I have more to tell you. So now when we look at, let me just make it a little neater. So when we look at what was happening in the electron transport chain, so let's pretend here are the proteins. And you have the oxidized electron carriers. Right? And so the electrons enter in, and then you have your oxygen down here as your final electron acceptor. Then you have your ATP synthase. That's going to, ADP plus inorganic phosphate is going to, as the protons flow through ATP synthase, it turns and it phosphorylates and makes ATP. Right? So we have two different things happening here. So this area is called electron transport chain. The ETC. And then this part, the diffusion of ions, is chemiosmosis. But together, this process is called oxidative phosphorylation. And I think you can remember this in maybe two ways. Either that these electron carriers had to be oxidized in order to power and have the whole thing happen, or you can think about like we had to oxidize our food in order to get these electron carriers reduced in the first place. Also, oxidative phosphorylation is going to produce a lot of ATP, about 30 give or take, maybe a little bit more, maybe a little bit less, for each glucose. And this is compared to the 2 that we got in glycolysis. So glycolysis only produced a net gain of 2 ATP. And then when we look at the citric acid cycle, 1 turn of the citric acid cycle only made 1. But it does happen twice because there's 2 pyruvates. Okay. Now the other thing too, my last thing I wanted to mention is that when we talk about these ions, they are like kind of like trapped, I guess, within this space because of the lipid bilayer. I thought that was important to mention. And inside here, the matrix, that is also where the Krebs cycle happens. So those carbon dioxides that were produced will diffuse out because carbon dioxide is non-polar. All right, so those are my notes on aerobic respiration, and